Alright, so the very first thing you're going to do, actually I shouldn't have zoomed out. The very first thing you're going to do is go on to google.com. Alright, so once you're on Google, you're going to search WSSP and click on the very first link, the Waxman Student Scholars Program. And then you're going to go to Resources and you're going to download Finch TV. 4 Peaks is the Mac version of it, but Finch TV still works on Mac, and Finch TV is better. It is superior. 4 Peaks just kind of looks nice, but you can do a lot more with Finch TV. So we're gonna go download Finch TV. You're gonna click it, and then you're gonna click on this link right here. Download it today. You're gonna fill all this out. You don't have to write organization and stuff, all that. Just write this bit, right? And then click submit. Write as much as you can and then click submit, and we're good. Right. And then once you download it, Finch TV kind of looks like this right here. There's a Finch and there's a TV. Get it? Finch TV. It's pretty self-explanatory. This is what it looks like. There's all these fancy things that you can do with it, which you'll learn eventually. I've also downloaded 4 Peaks just so you get to see what it kind of looks like. So this is 4 Peaks. Whoop-de-doo. As you can see, Finch TV seems to have a lot more stuff going on than 4 Peaks does. You can download either one. I personally prefer Finch TV, and you can do more with Finch TV anyway, and I'm going to be using Finch TV, so whatever works for you. And then you're going to go on to DSAP. Alright, you're going to go on to DSAP, you're going to go to Google. Why is it doing this to me? Okay, Google, and then you're going to you're going to type in DSAP 2015-2016, and click on this. Log in with your username and password. We all set it up in class. And you're going to come to a page that looks like this. This isn't my page, but I have all my clones done, so I can't really teach you on my page. It says continue. It's going to look like this. Analyze and assign to student. It says continue because this is the second take. No, actually third take on me trying to do this. Oh, please, no. Okay. Oh, thank God. Okay. Alright. Oh, it's still... Okay. Well, alright, we're gonna... Let's go back to welcome. Okay, you're going to click download for sequence. And it's gonna say, if you're on Mac, it's gonna ask you to do this. And you're gonna go on, and you're gonna open it. And it automatically opens on Finch TV. I'm going to open one on 4 Peaks, just so you kind of get the feel of what it looks like. I'm just going to drag and drop. Why can't I drag and drop? Okay. Oh boy. Okay, so this is what 4 Peaks looks like. It's pretty much the same thing, but like you can see, it kind of looks a little smoother because, you know, it's meant for a Mac than this does. But it's pretty much the same thing. You can use whatever you want. I'm going to use Finch TV because I kind of like Finch TV. And because I'm kind of obsessed with Darwin, and he has a thing about finches, so we're not gonna talk about that. You're gonna click Start Now, and this apparently has my progress saved from the last 3 trials. So is the 4 sequence readable? Yes, it is. This is what an unreadable sequence would look like. Hold on. Okay, why is... Alright, hold on. This is what an unreadable sequence looks like. You can't tell There's so many ends up here, you can't tell if this is 1 peak or 2 peak or 3 peaks. That doesn't even look like a peak, honestly. And then it starts plateauing here. What even is that? So this is unreadable. This up here is pretty readable. You can see each and every single peak. You know, it's very distinct. There aren't a lot of ends up here. Ends is unidentifiable base. I mean, that's not the official name, but I just call it that because it's an N that stands for a base that the machine can't recognize. So it's going to ask you where the sequence starts. The magical bit that you look for in every single one of your clones is going to be cat, tac, GG, CC, and then a run of G's. So basically cat, tac, GG, CC. Okay, this is what you're gonna look for, and then there's gonna be a bunch of G's running after the CC, and it doesn't matter how many G's. That's what you're gonna look for. And that bit is right here. Cat, tac, GG, CC, and then a run of G's. Your sequence starts exactly after this little run of G's at this A. Down here you can see what base number it is, and I write it in here, A63. In the unedited sequence, which is the last base. So for the last base, you're going to go, oh sorry, I scrolled way past. You're going to go to the poly A tail. Now if you remember when I was doing the lectures, mRNA has a poly A tail at the end of it. When we do cDNA, it's going to copy the mRNA, and you're going to have that exact same strand over here in the form of cDNA. So this poly A tail is still in the sequence. So you're going to take your last base pair is going to be this very last A in the poly A tail. And again, you look at the number of base it is at the end, and that's your number right here. And you're going to, you're going to select all of it. Come on. Don't do this to me. You're going to select all of it. Okay, we're gonna make my life easy. You're gonna select it all. You don't have to do that, I just did that so the scrolling would be quicker. And finding the stuff is so much harder now, oh my god. Oh right, A63, haha. So you go up to A63, you're going to copy the sequence, and you're going to paste it. Now, if you don't have the right number of characters, base pairs, it's going to call you out on it. Hey, look at that. The length of the sequence doesn't match your answers in questions 2 and 3. So you're gonna go back. You have to make sure that you put in the right amount of base pairs, and then you can pass. So in this sequence that you've selected, you're gonna look for, okay, let me make this normal again. You're gonna look for the letter N. There's a letter N, so you're gonna click on it, and you have to make the judgment now on what letter you think this is. All the A's are green, all the T's are red, all the G's are black, and all the C's are blue. This looks like an A to me, so I'm gonna click A and leave it in lowercase, because you want to be able to make sure, you want to be able to tell which ones you've changed and which ones you haven't. Leave the big A's alone, don't do anything to them. Change all of the N's. In the poly A tail, usually all of the N's are, well, A. And you're going to click it all again. Select it all again, I mean. It's going to take forever because Finch TV likes to do that. Am I on my account? Whatever. Where'd it go? Right here. Okay. So you're gonna copy this, and you're going to paste, oh boy. You're going to paste that here. Yes, you did make changes because there were ends in here. If you don't have any ends, which for this PC practice clone, you're going to have ends because we all have the same practice clones. If you don't have any ends in your sequence, when you get your sequence, you're going to click no, and this box kind of disappears. Wait, hold on. See, the box disappears, and you don't have to worry about that. So we're going to go on. Oh, we're all going to have a blast running these blast searches. Get it? Sorry, my friend made that joke and I totally stole it. We're going to run a blast N. So, in the blast sequence, blast searches, you're going to take this sequence that they give you, which is the sequence that you pasted in the last box, and you're going to paste it here, and then you're going to scroll down and run blast. So this takes a while to run, so let's not waste time, go back and click blast N again, excuse me, but this time we're going to run it in the express sequence tag database. So this database only has only runs your sequence against the expressed sequences. So basically the part of the sequence that's actually expressed, that actually gets translated into proteins. And you're going to paste the exact same sequence again in the in the paste little text box thing, and then you're gonna run the blast again. And okay, our blast is done. So less than 40 right here, base pairs, is a very poor search. If you have your search results in less than 40, you can pretty much count that. You can pretty much say that your sequence was horrendous. Sequence matches were horrendous. 40 to 50 is still pretty much the same thing. 50 to 80 is when it starts getting like, okay, I guess you can consider it somewhat reliable, but I wouldn't really care much for 50 to 80 base pairs. And then 80 to 200 is kind of where you go, okay, cool, we're getting somewhere. And then in the red zone, above 200 is where you want to be, because that's, on point matches. All of these lines right here are matches. These are all of those lines right here. And then if you scroll even further down, it's all of those matches in even more detail. So this is our blast N, and this is the S database. Okay, we'll get to that later. So in the blast N, you're going to put down 3 different organisms. So this organism right here, you can't have another organism of the same, you can't have another sequence from the same organism. I'm trying to look for the same organism to tell you can't have this. Okay, never mind that then. Right here, these two, you can't put both of these as one of your search matches. You have to pick 3 different organisms. Fortunately, the top 3 right here are different, so we're going to use those top 3. So you're going to go down, and you're going to look. Okay, you're going to type in the accession number. So we're all gonna use shortcuts here. This bit right here is the accession number. You're just gonna copy it. You're gonna keep your arrow at the length and copy, because you don't want to click on the link. It's not really bad if you click on the link. It just takes you to the actual page. Get rid of that little dash line at the end, and that's your accession number. Definition is all of that right there, and you're gonna click that, and that's gonna be your definition. Now looking for the organism name gets kind of tricky. So right here, I'm pretty sure none of you know where the organism name stops. It starts here, but you don't know where it stops. So what you want to do is you want to just click on the little link, and it takes you to a page about the entire thing, and the organism name is right here. You can use that. So we're gonna go back. Over here, I kinda am able to tell the organism name because it's an acquired, what's it called? Skill. There you go, an acquired skill. After that, it's the query start. So there's, over here you can see, like, there's a subject and query. You see 61C. You're gonna paste in C61, and then the end, just look at the last one, T384, T384, and the E value, where the expect value right here. So this is basically 7 to the negative 46. The smaller your number, the better it is. So so the bigger this number right here, the bigger this number right here, the better it is. Because that means that the entire number after the exponent and after you've kind of, done the whole thing, is going to be smaller because it's 7 to the negative 46, right? So it's going to be an extremely tiny number. Not too tiny, but like pretty small. So the smaller your expect value, the better the match. You're just going to paste all of these things in there. Then you move on to the next organism. Again, you take the accession number and then the definition. So yeah, you probably should be doing this with me. Ooh, what did I type? G383. And the last E value. Alright. So we ran the second blast. What we're gonna do is take this out and we're gonna minimize it because I keep all the blast searches on because you eventually end up needing one of them again, and then you have to run it all over again, and then it starts getting frozen in the middle because everybody else is running their blast searches at the same time, and it just gets really annoying. So just after you run them, leave them on until you're done with the whole thing. That way everybody is happy. So this is your blast search in the EST database, EST, Express Sequence Tag database. So you see how all of these matches are super duper good. They're over 200, so they're incredibly amazing. This looks like the exact same match. 200. Okay, so the expect value is 0, so it's the lowest you can get. It's an exact match. So yeah, it's pretty good. You're going to want to put that in there. And hey, look at that! It is duckweed. What we're searching. Unfortunately, all of these little red matches are all duckweed. You can't have 2 duckweed sequences in your match list. So you're gonna have to pick another organism right here. This is where it starts from a different organism. And then this, I assume. Nope, that's still the same organism. Nope. Yes, this is a different organism. You end up figuring it out. And if you can't tell, again, you click this and you go look under the organism name. Once again, you're going to do the same thing. Grab the last few digits. That's going to be accession number. Definition is that first entire line. Organism, if you can't tell, it's duckweed or Landoltia punctata. We're going to use the official name because official names are cooler. And the query starts at A1 and ours starts at A1, so it's a perfect match. ends at, what was it again? 727. Wait. Yes, A727. And the e-value is 0.0. My very first year, I was kind of confused with an e-value of 0.0. I honestly had no idea what to do with that. I didn't realize 0.0 was the lowest number. I don't know why, but I didn't realize it, and I literally sat there staring at the screen for like half an hour wondering what to do. Okay, wait. Okay, this is not duckweed. Yep, okay, so next organism that isn't duckweed. T379. Okay, nothing is typing. T379, unless it was typing somewhere else and I missed it. Oh, okay, never mind, it wasn't. T379. Alright. And the expect value. I swear this computer has a mind of its own. Okay. Is your edited sequence significantly similar to sequences found in any other organism? Okay, well, this is a different organism. It seems to be significantly similar, so I'm going to say yes. Look down at the list of blast end results. Is your edited sequence significantly similar to sequences found in different kingdoms? These are all plants, so I'm going to say no. Oh. Hold on. How do you tell if these are all plants, right? So, I told you blast end was going to come in handy. You open up your blast end again, scroll up so you're kind of over here. You're going to go to the first page. Okay, fine. You're going to go over here, open it. Okay, right here. You have your domain, kingdom, plants. Look at that. Okay, instead of clicking on the name, let's click back here. Haha, I figured it out. Same thing. And again, on the last one, yep, it's still the same thing. So, no, they're not in different kingdoms, so that's why it's no. Then you move on to Blast X. Blast X is the most important blast. It runs your sequence against other sequences, but not just nucleotides. It runs it against proteins too. So it kind of puts them into codons and runs it against a protein. I don't know if I make sense. Dr. Lawlin definitely explains the blasts better than I do. So I'll most likely have him explain these in class. So yeah, we'll do that. So this is gonna run. Take this out, and we're gonna minimize this. We're gonna minimize this. Look at this. It's gonna start showing you what your sequence is most likely coding for. So your sequence, the PC clone, not PC clone, this clone right here, it matches most closely to the thyrodoxin superfamily. Thyrodoxin is one of the most common proteins that we find. So if you get thyrodoxin as your protein, don't be bummed out, but be bummed out a little bit, because that's an annoying protein. It kind of just means that, hey, you just found something that everybody else has been finding for like eons now. I'm sorry, I apologize for being rude like that, but if you find thyrodoxin, you've basically found nothing. It's a new sequence, don't get me wrong, you can still publish it, but thyrodoxin shows up everywhere. But this probably tells you that thyrodoxin plays a very important role in the plant because it shows up so many times. It's such a commonly found protein that it's bound to have an important role. And we'll figure out what exactly it does as we analyze this and why it's such an important role. If it has an important role. I had thyrodoxin. I totally forgot what it did. I'm so sorry, Dr. Lalin. If you're gonna watch this, I apologize. Okay, so you're gonna do the same thing for Blast X. And hey, look at that. There's Blast P, Blast X versus Blast P. We're gonna do this for a while. So again, you scroll all the way down, do the exact same thing. But if you see, this isn't proteins. These are all of these letters, they stand for proteins. They're not nucleotides anymore. So Blast X runs your nucleotide sequence, turns it into a protein sequence, and runs it against other proteins. So this is, you know, a good thing to rely on. Also, these little plus signs right here, that just shows that even though this K and the R, even though those two proteins aren't the same protein, they have similar properties. So they behave in a similar fashion. For example, both of them could be hydrophobic. So they could both be, you know, tucked inside the protein and still make the protein look similar. And as we all know, in the world of science, structure equals function. So if a protein is similarly structured, it's going to have a similar function. Or if it has a similar function, it's going to have a similar structure. It goes either way. Anyway, back to Blast X. Copy the accession number. Paste the accession number. This is always fun. Okay, so Blast X kind of makes finding the organism name easy because it's usually in these brackets. So Blast X is kind of going to give you a break. Query start M72. Query end L380. 380. E value is decent. Okay, move on to the next organism. Okay, so we see that all of them start at M72 and L380. All of them. And, you know, they all start around the same, what's it called? Mark. So, we can assume that this bit right here is what codes for this thyrodoxin, whatever this protein is. Thyrodoxin-like protein, there you go. So we can guess that in this little, I'm like doing things with my hands, like pointing with my hands like you guys can see. But this right here, this portion right here is most likely the coding region for, the expressed region for our sequence. That's probably where the thyrodoxin-like protein is being coded for. So once we have all of this done, we answer these questions. Yeah, wait, hold on, we forgot the, hold on, what organism was that? There you go. You can get all that stuff from over here too. This is the accession number, this is the E value. The only thing you can't get from here is where the query starts and ends. So I kind of just scroll down and get it from there because everything is there. Is it significantly similar? I would say so. Is it found in different kingdoms? I don't know, actually. Open link in new tab, open link in new tab, open link in new tab. Alright. Oh my god, nope, I clicked the wrong stuff. Open link in new tab. I swear to god, this computer is going to make me throw it out the window. I actually shouldn't be saying that. I have a privilege. I apologize. Nope, plant, plant, and plant. Alright, so they're all in the same kingdom. No. Keep going. Now we're gonna go to Toolbox. Take this out and minimize it. Keep them all open. See? Keep it open because it's gonna come in handy eventually. Actually, I'm gonna minimize this too so it's not a headache. Alright, so this is what Toolbox looks like. You're gonna go in, retrieve your sequence, and you are going to paste here. Now you're gonna go back to BlastX. I think this is BlastX. Yep. And you're gonna scroll all the way down to here. At the end, it's going to tell you if your sequence is in frame 3, 2, or 1. So basically what these frames say is where your sequence starts. So it could be AGG, and then it's gonna go every 3 from there, or it's gonna skip the first one. It's gonna go every 3 from there, right? Or it's gonna skip the first 2, and it's gonna go every 3 from the third one. And there can only be 3 frames because there's only 3 codons. So if you skip the first 3 and go to the fourth one, you're essentially starting at frame 1 again because AGA right here was part of the first frame. So our BlastX is going to tell us which frame we're in. If you get multiple frames, something is off. If you get negative frames, something is definitely off. But we're in frame 3, so you're going to look for the longest sequence in frame 3 and start with the M because all proteins start with methionine. I hope I said that right. Methionine. Yes, I did. Methionine. Okay, just making sure. You're going to start from M, go all the way down to the last letter. Stop codon is not part of the actual protein, but it is part of the sequence. You're going to put which frame it's in. If you get a negative frame, you're going to put none. Paste the most likely protein sequence or portion of protein sequence here. Okay, so here's the thing. It could be from here to here, or there could be another M way before this letter right here, and we wouldn't know about it, so this could be a partial protein. But how do we tell if it's a partial protein or not? Well, we can tell because we have our handy dandy BlastX right here. So the subject, the query is you. You are the query. And the subject, well, you're not query, but your sequence is the query. And the subject is the other sequence. So because the subject starts at 1M and yours is 72M, well, yours doesn't really matter. But because the subject starts at 1M and not like 32M, you can see that it's your M is the first M of all of the sequences. They all start at 1M. They're all starting at 1M. And they're ending at 103L, right? So you can assume that this M right here is your first M72. Well, not M72. This A72 is where it starts. So you're gonna select everything from M up to L. You're not gonna select the stop codon. This little star right here is a stop codon. You're not gonna select it. You're gonna copy it and you're gonna paste it. At which base does the open reading frame start? Okay, so this is your open reading frame, what we just selected, alright? This right here It starts at A72. So you're going to click A72. Ooh, that's 73. And at which base does it end? This is where you want to include a stop codon, because even though the stop codon is not part of a protein, it's still part of your open reading frame. The, what's it called? The ribosome needs to read the stop codon for the protein to stop being made. I really hope I make sense. So you're going to take the last base here, G383, and you're going to put that here, 383. And next. And now we're going to run a blast P. You just leave the toolbox on too, because why not? Leave everything on. Don't turn anything off, ever, in your entire life. No, I'm sorry, that last bit, disregard that. Oh boy, I'm running out of battery. I would cry if this kind of died in the middle. Okay, so what was I saying? Blast P, right. We're gonna run a blast P. You're gonna retrieve the protein sequence, and now it's just gonna run protein sequences against other protein sequences. We're gonna run blast P. I'll actually call it a day after we run all of these blasts. We'll do analysis some other day. And once again, we wait for these blast results to load. I think this is pretty much it for the blast. Yeah, this is the last blast search. Okay. Blast P. We scroll down, we do the exact same thing. Why do you have to do this to me? M1, L103. Please be the same E value. Yes. Well, I mean, okay, I'm already here. Wait, okay. We have to look for other organisms. Note the repetition of the same 3 organisms. This happens because these 3 organisms are probably very closely related to Landoltia punctata, and they produce very similar proteins as the plant Landoltia punctata does. So, you know, you kind of gotta start thinking about this stuff. You don't just, you know, copy and paste for the sake of copy and pasting. You gotta notice things. Bring out your inner Sherlock Holmes. Oh wait, they're all the same. Please be the same. Okay, good. Oh, it's not. Look at that. That demonstrates poor work ethics when you just want to copy and paste and you see that the answer is not correct, because I would have just copied and pasted and assumed it was the same because I'm a reckless person. See, so you kind of gotta make sure, okay? I'm kidding, I'm not a reckless person. Does your DNA sequence code for the likely start of a protein? Yes, I do think so. Why? Because, again, they all start at M1, and it's not subject doesn't start at what, like 60 something, or some random number in the middle of nowhere. Does your DNA sequence likely code for the end of a protein? Well, yeah, because they all seem to be ending at the same place. So yes, it does seem to be coding for the end of a protein. Oh god, sorry. Alright. Are any of the proteins found in BLAST-P search the same as any of the proteins found in BLAST-X? So you match up the accession numbers. This is the exact same thing as this. Look at that, this is the exact same thing as, oh yes, they're all pretty much the same thing. Compare the E-values for the same proteins. Are they similar? Yes, they are. Similar meaning, are they like 1 to 2 numbers apart? Yes, they are. Compare the start and stop positions for BLAST-X and BLAST-P alignments for the same protein. Are the alignments the same? So for this, you're kind of gonna have to go back. M72, L380. The main point is starts at M, ends at L. What? Okay, this is weird. Yes, L380. BLAST-P. Whoa, why is it doing this to me? Why do I have to start over again? I don't understand. Are the alignments the same? Yes, they are. Save. You know what? It's okay. I'm not gonna worry about it. It's okay. You can say L380 as much as you want. Okay. So this is the tricky part. You're gonna go back to Toolbox because Toolbox is easy to use. Well, a lot easier than what they give you in DSAP. So this is your open reading frame, right? This up here is the 5' UTR because, you know, it goes from 5' to 3', not the other way around. So here's your open reading frame, right? And here's the 5' UTR. But like I said before, you can see that it starts at G3, not the very first base. So what you do is go to frame 3, well, stay in the frame you are, and then you select it and just take the end. So C71, and you're going to put in the end C71. I'm going to take this out because I made errors. See, this is my reckless side coming out. And then you go to frame 1 and just kind of highlight the very first protein, and it's the very first base pair is A1. So here's A1, C71, A72, G383. So the next one is going to be something 384. Go back to frame 3, and the one right after stop codon, T384. What was it, 384? And the very end, which is what? A728. Now, the not applicable stuff comes in if, let's say that this M wasn't the start of our protein, and the start of our protein was somewhere, you know, before this bit right here, then we would say that the 5' UTR does not exist. Or if we never reached the stop codon, and let's say it was something like this, and your protein started over here and it just kept going and there was no stop codon, then you would say that the 3' UTR does not exist. But it does. See? Right here. You have all of this and we're happy. Okay, I'm gonna call it a day at this because analysis is a whole different world. This is where you kind of have to actually think and use your brain. So we're gonna do this another time because I got through the nitty-gritty stuff. Just look over the video. Well, I mean, this is at the end of the video, so I can't really, say anything. But if you guys did it along with me, just make sure you click this Save Now button. Go back, just click Save Now, and you can log out and whatever. But now you can turn everything off. If you want to carry out your own analysis and go on without me and be a brave little soul, what I suggest doing is opening up, what is this, Blast X? Blast X, okay, I suggest opening up Blast P. Is this Blast P? Yep, this is Blast P. Okay, you're gonna open up Blast P. You're gonna click this. You're gonna click this little blue thing. And it's gonna take you to a place like this. And you're going to hover over this ooh. Sorry, I hit myself by accident. You're gonna click this dark blue, well, not click it, just hover over it, and it's gonna show you all of these things. Read this carefully, it's gonna give you all the answers you need for all of these questions. Leave this one alone because this you might not know how to find. Although I guess I can tell you, you're gonna click on the first one, open link in new tab, and you're going to see if it exists in humans. Right here, you're gonna click the very first one. Organism, no. Try click on the second one. Organism, oh look at that, it does exist in humans. Okay, I should probably stop because now I'm carrying you through the analysis. But if you feel like a brave little soul, try to go as far as you can in analysis. I'm pretty sure you guys won't know what half of these things mean. But do all of this, and yeah, you guys should be fine. I hope you had fun running all of those. No. I hope you had a blast running all of those blast searches. I've been waiting so long for you to use that. Like, you have no idea. My friend told me it. I wanted to use it for so long. But yeah, glad you got all that done. Click Save Now as many times as you can because there was a time where I did not click Save Now. It was horrendous. And you're just gonna log out. Wait, no. Go back to my clones page. And it's gonna say being worked on by student, and you're just gonna log out. And there you go. That's the end of that. You can turn everything else off. You can turn off. I'm so sorry. I keep hiccuping in the middle. Do I want to save this file before exiting? No, I don't. PCs right here. I have a little folder for everything. You can turn everything else off now. And you guys can be happy little DNA analyzers. The end. Good night, guys. Or have a nice day, whenever you decide to watch.